So today we'll see the preparation of various media for bacterial growth. So culture media is defined as any material that is prepared for the growth of bacteria in the laboratory. So bacteria can be grown in synthetic or defined media, that is medium in which all the components are known as termed as the synthetic or defined media. Examples are many chemoorganotrophic heterotrophs. They are grown in the defined media with glucose as the carbon source and ammonium salt as the nitrogen source. So these defined media, they are widely used for research purpose as it is desirable to know what the experimental microorganism is metabolizing. Next is the complex media. So media that contains some ingredients of unknown chemical composition is the complex media. Such media are very useful because these single complex medium is sufficiently rich to meet the nutritional requirements of many microbes. Complex media contains undefined constituents like peptones, meat extract and the yeast extract. So now we'll come to the types of media. Selective media. Selective media, it favors the growth of one organism over another. Bile salts or dyes like the basic fusion and the crystal violet, it favors the growth of gram-negative bacteria, thereby inhibiting the gram-positive bacteria. Then the differential media. So this differential media, it distinguishes between the different groups of bacteria. For example, blood agar, McConkey agar, etc. Blood agar, it distinguishes between hemolytic and the non-hemolytic bacteria. While McConkey agar, it contains lactose and neutral red dye that differentiate the lactose fermenting colonies that appear pink to red in color from the non-lactose fermenters. So we'll see the preparation of nutrient broth first. In the principle, nutrient media, they are the basic culture media that are used for maintaining microorganism and cultivating fastidious organism. Nutrient broth is one of the several non-selective media that are useful in routine cultivation of microorganisms. It can be used for the cultivation and enumeration of bacteria which are not particularly fastidious. Peptic digest of the animal tissue, beef extract and the yeast extract, it provides the necessary nitrogen compounds, carbon, vitamins and also some trace ingredients that are necessary for the growth of bacteria. Sodium chloride, it maintains the osmotic equilibrium of the mediums. In the requirement, peptone, beef extract, sodium chloride, distilled water, and the pH should be maintained at 7. Then measuring cylinder, autoclave, hot plate, cotton, and so on. Next we'll come to procedure. Weigh amount of peptone, beef extract and sodium chloride are added to the conical flask containing 100 ml of the distilled water. Then the flask is heated with agitation to dissolve the constituent. Further, the pH of the medium is adjusted to 7 using a pH meter. Then the flask is sealed by the use of cotton plugs. Then the medium is autoclaved at 121 degrees Celsius for 15 minutes at 15 pounds of pressure. Then the medium is allowed to cool and is stored at room temperature for future use. In the precaution, cotton plugs are to be kept loose during autoclaving. Then the medium is to be stored at low temperature in the dust-free environment. So now we'll see the preparation of nutrient agar. So nutrient agar is a general purpose media, and a nutrient medium, it is used for the cultivation of microbes that supports the growth of a wide range of non-fastidious organisms. Nutrient agar is popular because it can grow a variety of types of bacteria and fungi and contains many nutrients that are needed for the bacterial growth. Nutrient agar is composed of peptone, beef extract, sodium chloride, and agar. So this peptone is an enzymatic digest of animal protein. Peptone is the principal source of organic nitrogen for growing bacteria. Beef extract is the water-soluble substances which aid in the bacterial growth, such as vitamins, carbohydrate, organic nitrogen compounds, and salt. Then the presence of the sodium chloride in the nutrient agar maintains a salt concentration in a medium that is similar to the cytoplasm of the microorganism, and agar is the solidifying agent. So this nutrient agar, it is frequently used for isolation and the purification of cultures. It can also be used as a means for producing the bacterial loans that are needed for antibiotic sensitivity test. So in the requirements, peptone, beef extract, sodium chloride, agar, distilled water, and the pH to be maintained at 7, then measuring cylinder, autoclave, hot plate, cotton, etc. In the procedure, weigh amount of the peptone, beef extract, and sodium chloride are added to the conical flask that contains the 100 ml of the distilled water. Then the flask is heated with agitation to dissolve the constituent. Further, the pH of the medium is adjusted to 7 using a pH meter. Finally, weigh amount of the agar is added to the flask. Then the flask is sealed by the use of cotton plugs. Then the medium is autoclaved at 121 degrees Celsius for 15 minutes at 15 pounds of pressure. After autoclaving, the medium is allowed to cool at approximately 45 degrees Celsius, but it should not be solidified. So this nutrient agar media is poured on the sterile petri plate and it is allowed to solidify. Today, we'll perform the different techniques for culturing bacteria. So microorganism can be isolated from various sources such as soil, air, water, sewage, decomposing matter, body surfaces, etc. And microorganism can be grown in the laboratory by providing its optimum conditions that are required for their growth. So, a culture that contains only one type of microorganism is known as the pure culture, while the culture that contains more than one kind of microorganism is known as the mixed culture. So this pure culture is maintained in the microbiological laboratory to study the colony characteristics, biochemical properties, morphology, staining reactions, immunological reactions, etc. The three most commonly used methods of obtaining pure culture of microorganism are the streak plate, pour plate, and the spread plate. So now we'll see the streak plate method. The streak plate method is used for obtaining discrete colonies and the pure culture. So this method was developed by Loeffler and Gefke. In this method, a sterile inoculation loop dipped into a suitable diluted suspension of organism, which is then struck on the surface of the solidified agar plates to make a series of parallel non-overlapping streaks. And the main aim of this technique is to obtain the colonies of microorganisms that are pure. So now coming to requirements. The first one is 24 to 48 hours nutrient broth culture, then the nutrient agar plates, inoculation loop, spirit lamp, marking pencil, etc. In the procedure, petri plates containing nutrient agar medium are labeled on the bottom with the name of the organisms to be cultured. Then, inoculation loop is sterilized by heating in the flame and after cooling, 1 loopful of the bacterial culture from the tube is taken. Then, the pretty plate cover is lifted by the use of the left hand and holding at 60-degree angle. The inoculum is placed on the agar surface and the inoculum is streaked from side to side in parallel lines across the surface of the area. Then, the inoculation loop is reflamed and cooled, and further turning the petri plate at 90-degree angle, the loop is touched at the corner of the culture in area 1 and the inoculum is streaked across the agar in area 2 as shown. It should be noted that the loop should never enter the area 1 again. Then the rest of the agar surface is now ready to use for complete streaking. Then the lid of the petri plate is replaced after completing the streaking and the loop is sterilized again by flaming. Then the plates are incubated at 37 degrees Celsius in an inverted position for 24 to 48 hours. And in the results, the plates are examined after incubation for the bacterial growth. A confluent growth is seen where the initial streak was made and the growth is less dense away from the streak and the discrete colonies farthest away from the streak. Today we'll see the pour plate technique. In the pour plate technique, serial dilution of the original specimen are added into the sterile petri plate, to which is poured, melted and cooled at around 45 degrees Celsius agar medium and is thoroughly mixed by rotating the plates and is allowed to solidify. After incubation, the plates are examined for the presence of individual colonies growing throughout the medium. Further, the pure colonies, which are of different shapes and size, may be isolated into the test tube culture media for obtaining pure culture. This technique is used as a means of determining the number of viable organisms in a liquid, such as water, milk, etc. In the requirements, 24-hour nutrient broth culture is required, then culture tubes, sterile 9 ml water blanks, sterile petri plates, water bath, spirit lamp, marking pencil, etc. Then in the procedure, first thing, serial dilution of the sample is done. Then 1 ml of the bacterial suspension is transferred to the labeled petri plate. Then the lid of the petri plate is slightly lifted with the left hand and the sterile molten agar is poured on the plates. Then the dish is rotated gently to mix the culture and the medium thoroughly and to ensure that the medium covers the plate evenly. Further, the medium is allowed to solidify and it is incubated at 37 degrees Celsius for 24 hours. In the result, lesser number of colonies are observed in this test and in this technique as compared to the strict plate method. Further, the colonies may be subcultured for future use. Now we'll see the spread plate method. Spread plate technique is used for the separation of a dilute mixed population of microorganisms so that individual colonies can be isolated. In this technique, microorganisms are spread over the solidified agar medium with a sterile L-shaped glass rod while the petri plate is spun on a turntable. The main aim of this technique is that the cells will be separated from each other by a distance sufficient to allow the colonies that developed to be free from each other. In the requirements, 24-hour nutrient broth culture is required, then nutrient agar plates, L-shaped glass rod, beaker, spirit lamp, marking pencil, etc. into procedure. The nutrient agar plates are labeled. Then the L-shaped glass rod is dipped in the 95% ethanol. Further, aseptically, a loopful of the culture is transferred in the appropriately labeled nutrient agar plates. Then the glass rod is removed from the beaker and it is placed on the Petri plate. The glass rod is gently pressed and rotated back and forth for spreading the culture over the agar surface. Then the glass rod is immersed in the alcohol again and then it is reflamed to sterilize it. Finally, the plates are incubated in the inverted position at 37 degrees Celsius for 24 hours. In the results, the plates are examined for bacterial growth and some of the colonies are free from each other. The discrete colonies that are formed can be used for further study. So that was about the media for the growth of bacteria. And to sum up today's topic, bacteria, in contrast to fungi, are often cultured in a media that are used in the routine bacteriological laboratory, which is composed of the constituents like the beef extract, which is a derivative of beef and is a source of organic carbon, nitrogen, vitamins, and the inorganic salts, than the peptone, which is a semi-digested protein. Hence, for the successful cultivation of a given microorganism, it is necessary to understand its nutritional requirements and then supply the essential nutrients in the proper form and proportion in a culture medium.